Hello learners, welcome to NeenvIAS. This is our concept behind current affairs series. My name is Kshitiz Dhiman and the topic today is CRISPR Cas9. Genetic scissors, it is also known as, right? So let us start with this session, and first of all, since this is a concept behind current affairs, so current affairs will be discussed, but at the end, because what was done in that current affairs, I need to explain the basics first, and then you will be able to relate to it properly, right? So with that understanding, let us start. So briefly, let us understand what is this technology. Just briefly, just an introduction to some of the terms, because there are a few technical terms. So let us introduce them one by one, right? So it's a gene editing tool. What is CRISPR? What is CRISPR Cas9? It's a gene editing tool, all right? For example, let's take an example of Microsoft Word. It is a word editing tool. Right? Now, if we consider genes, it is CRISPR Cas9, and if we consider, let's say, simple document, it is Microsoft Word. So in Microsoft Word, you can go write something, delete something, right? So you can understand how to create an essay or a small paragraph. In the similar manner, genetic engineering by Cas9, CRISPR Cas9 is making all these things possible. For example, you can turn gene on and off. You can, of course, change its behavior. We'll see everything. So just to understand, it's a gene editing tool, like Microsoft Word is a word, let's say, text editing tool. But one of the fantastic thing is, it is derived from nature. Humans have not developed this process. They have captured it or derived it from nature itself. The whole thing is done by nature, right? It's an immune response in bacteria and other organisms. So CRISPR can be used to change the DNA, right? And it can turn genes off and on, like I just discussed, correct? So this is the introduction. Now we are going to see one by one the technical terms and understand their meaning. Then we'll see the process, okay? So first term that we are going to see, it's CRISPR itself. Let us see the full form first. So CRISPR stands for clustered, regularly interspaced, short, palindromic repeats. Now if you see this term for the first time, it is a mouthful, and of course there are very different terms, and basically Shashi Tharoor level English is also there, okay? So let us understand them one by one. I have brought some examples for difficult words also. So the first word is clustered, right? Clustered means grouping together. For example, just imagine bunch of grapes or bunch of bananas, they are sticking together. So similar way, it is clustered, right? This word means that. So what happens is, in DNA, CRISPR, the CRISPR term, not Cas9, the CRISPR term is a collection of specific DNA sequences. We'll see the diagram also, then you'll understand, okay? That are grouped together in a particular spot in an organism's DNA, right? Next term is regularly interspaced. I think you all would know what is regularly, okay? So it happens at fixed intervals, and interspaced means, inter means between two things, space, of course, there is some gap. So they have spaces between them. So CRISPR's DNA also have regular spaces between them. So one thing is clear from this discussion, that CRISPR is part of the DNA, right? Next term, short, of course, very easy to understand. So DNA sequences in CRISPR array is short one. We'll see the diagram. Palindromic, this means something which is similar. When you read it, it's similar from the start and the end, like madam or race car. If you read them from the front, I have the example, yes. So M, and D is in the middle. So the word is similar, is not similar, it's exactly the same from the starting and from the end. Last word is repeats. Of course, this means it happens again and again. So in a song we have chorus, right? We have chorus. It repeats again and again. So similarly, this also repeats, right? Next thing, in CRISPR, you need to understand, like I've already mentioned, that it is a section of DNA sequence. We are clear about this, okay? So it is also known as CRISPR array or repeat spacer array. Now, why am I telling you this? Because UPSC is in a habit of asking synonyms and confusing you. See, they cannot ask very difficult questions in general studies paper with regards to any subject. All right, there is a limit to which they can go, otherwise it will jump into the optional category or it will go too much technical in science and technology. So they will ask different things, but they will ask the same concept, right? So these synonyms are important. So what is it made of? You should ask yourself such questions also whenever you are studying some topic. So it is made of DNA of invading virus. I've told you that it's an immune response. So right now we are trying to understand how nature does it. Then quickly you will be able to understand how scientists developed it and borrowed it. Correct? So this is important. So it is made of DNA of the invading virus. We'll see the process. It has two components, repeats and spacers. Spacers here is more important. Spacer means it keeps the information of the invading virus, and basically it takes the DNA of the invading virus and keeps it with itself. Okay. So I've explained spacer. Also, it's a genetic memory. That means it is passed down through generations of bacteria, right? Like immunity is passed down in humans and genetic diseases are also passed down. So similarly, this is also, right? So spacer, I have also mentioned what it is. It is from, spacer is DNA sequence from the virus. So it is basically a photograph of virus's DNA, and basically for bacteria, if we break it down or make it more simpler, spacer is a photograph of the virus. By this photograph, the bacteria is able to identify if bacteria is attacking, bacteria is able to identify if the virus is attacking again. Okay. So permanent genetic record, right, of past infection is created in CRISPR array. This is important. This statement is important. I am trying to bring different perspectives so that you understand how to deal with this topic, right? So remember this. Although it might sound a bit technical, but I think we have discussed every term. So this is how CRISPR DNA looks. There is viral DNA in it and there is repeated sequences. So this is spacer, right? This is the repeater. So you can see it is in different color. The viral DNA is in different color. Why? Why? It is because it is from different viruses. Okay. And what is the role of repeater? It is basically a boundary. That's all you need to understand to separate the different genes of the virus. So it is called CRISPR array. This whole thing, repeater and spacer, is called CRISPR array. Okay. Now we come to the next term in CRISPR Cas9 term, which is Cas9. So Cas is basically CRISPR associated. Cas means CRISPR associated, and 9 stands for the protein 9. Now this is also derived from nature. This Cas9 thing is also derived from nature, right? Now what happens is it uses the RNA. Okay, we are talking about natural process. So the bacteria, let's say the bacteria is being attacked by the virus. So bacteria will develop an RNA to find the virus. All right. So Cas9, the protein, will follow the RNA to find the virus and attack it and break its DNA so that the virus cannot replicate, right? This cutting action is used by bacteria for defense, which I just mentioned, right? So scientists have adopted this and they are using Cas9 to cut the genes of various organisms deliberately, right? Not as an immune response, but using it to target specific genes and cutting them. Okay. So this is also called as molecular scissors or genetic scalpel. scalpel is sort of a blade used by surgeons. So you can understand again why am I telling you the synonyms. This is important. Okay, let's move forward. There are types of the proteins which cut the DNA of anything, right? Any organism. Cas1 is there. This is 9, right? So there is Cas1, Cas2, and other are also there, but we are not going to discuss them because that is not our purpose. You need to understand that Cas, CRISPR associated protein 9, or Cas1 or Cas2, these are scissors which cut the DNA, right? You need to understand that's all. All right. So Cas9 is the most widely used. It is easily programmable by the scientists so that it can cut the specific part they want to cut. All right. So easily programmable. It can bind to almost any desired sequence. And yes, one more advantage is there, that it can easily follow any RNA that is there. Right. So this is also important when you're trying to edit a gene in a person. You need to change the correct gene, right? Edit the correct gene. So you need to give a target, and of course, whenever you shoot something, let's say for example, Olympics are going on, and recently a person from Turkey won, right? So he was shooting. So of course, he won the silver medal because he shot the proper target without any stuff like, okay. So that is the point. You need to shoot and you need to cut the right gene. And Cas9 can be customized very easily also, right? So one thing we need to understand, which I've already told you, you can turn genes on and off, okay? And you can also change the behavior. You can reduce the effect of the gene and increase the effect of gene. So that is called CRISPR A and CRISPR I. We'll see that in detail soon, right? Other Cas9, CRISPR Cas9s are base editors. That means they just change one letter of the DNA to another, right? So this is how it can change. So it is used for genetic editing, and these are the different ways in which nature uses it. Let's move ahead. Third terminology, which is called PAM, which is protospacer adjacent motif. Now this word motif, it means a pattern, right? And proto means the first one, the original one, and spacer is the virus DNA. Adjacent means just beside, right? So let's say the board is adjacent to me, right? So this is what PAM means, and it is used in a very good way. Although if you read about CRISPR Cas9, most of the attention is given to CRISPR or Cas9, but PAM is very important. So what is the role, right? Of course, why it is important. So this viral DNA is stored in the bacteria's DNA also, like I've just told you. So in nature, we have taken this from nature, this process, and it is found in bacteria and archaea, right? So in nature, this thing, PAM, helps to identify which is the viral DNA that is supposed to be cut, because the CRISPR array also has viral DNA. Let me just show you again. So CRISPR array also has viral DNA, and virus of course has the viral DNA. So which one should be cut? That is told by PAM. In CRISPR array, you can see the diagram. There is no PAM or nothing I have labeled like that. But in the virus that is attacking the bacteria, its DNA, virus's DNA will have the PAM. Okay. Sorry, right. So how does the viral fragment is identified which is to be cut? Because itself the bacteria is having it, right? Solution is the PAM. Okay, it is basically a locator, the enemy's locator. You find it and you cut that DNA. That's all. It's the role. So it's a short sequence after the target DNA which is supposed to be cut. Okay, if the target DNA does not have PAM, it is not cut, and CRISPR array does not have any PAM, right? Now let's move ahead. So in the nature, how it works, let's see just few facts, although things might have been already told. It is an immune system that bacteria and other microorganisms use to defend themselves against virus. This is the proper definition, which I have not discussed till now, but I have explained everything. So CRISPR is found in approximately 50% of the sequenced bacterial genomes and nearly 90% of the sequenced archaea. So although we have sequenced a lot of bacteria and archaea and it is found in a lot of them, but it is a general thinking that it is available to all bacteria, right? Now let us see the process. Since we have understood the terms, we can now understand what is happening, right? So this thing is the virus which is attacking. It is also called a phage. All right. So it has a DNA. This is the DNA sequence. Now Cas1 and Cas2, they are used to cut the DNA sequence from the actual virus. They cut the DNA sequence from the virus and bring it to the CRISPR array of the bacteria, right? So this is the viral DNA. They have cut it. This structure is the Cas1 and Cas2. They bring it, they cut it out, right? This DNA is cut out and it is brought to the CRISPR array. So new spacer is added whenever bacteria has an infection. It takes the DNA of the virus and stores it for future immune purposes, right? Now what happens next? Let's say the reinfection is happening. So what will happen? This is the CRISPR array, all right? Now this is from different viruses. That's why they are in different colors. Repeaters are in same color. They have no effect on the virus DNA that is stored. So let's say the attack is happening again, the virus is attacking again on the bacteria. So what will happen is every spacer will get transcribed, that is basically written in simple language. It will get written in the RNA, right? Now that RNA then will be broken down into individual spacers so that they are different. Right now they are clubbed together, again just a small step in the creation of identifier RNA. So this is called CRISPR RNA. It is also called as guide RNA because of course it guides towards the virus, correct? So this is how second step is. Now in the third step, there is a surveillance complex. Complex means collection of things, right here. So RNA and the Cas9, this green thing is Cas9 and this is the RNA which has the virus identifier. This will go to the target. Let's say there is infection happening. So it will find the target and if this matches, red one matches, it will attach to it, and since Cas9 is the scissor, Cas9 will cut the DNA and virus will die. So what happens is, first from the virus we have take something, we identify it, we store it, and then if the reinfection is happening, we break it down, we break down the CRISPR array and then we again attack the virus, okay? So understand this, what is the role of CRISPR, what is the role of PAM? I'll show you PAM in the next diagram. So till now we have discussed the role of CRISPR array, we have discussed Cas9, we have discussed the guide RNA. Now in this diagram you will see PAM is labeled, all right? So this is the virus. Let me come at this side. This is the virus. This cloudy thing is Cas9, right? The genetic scissor. So guide RNA is there. It will identify the invading virus, and invading virus DNA will have PAM, right? So Cas9 checks for PAM and then guide RNA finds the virus and its DNA, PAM is checked and then Cas9 cuts it, right? So Cas9 cuts the DNA wherever the PAM is found, it will cut the DNA. As you can see, it separates it in two parts, right? So this is the clear diagram, a little more technical diagram, that's why I bought this. NGG, as you can see here, NGG. Now this is a sequence which is PAM of a virus, all right? So guide RNA finds it, This is the proto-spacer sequence. I hope you understand what is proto-spacer. This is PAM sequence. This is target DNA. This is of course the Cas9. So this is more technical, right? I'm not going to explain because we have done everything. All right. So these are the steps. I've already discussed everything. You can pause the video and read it for your benefit. I have clearly outlined the steps. Now, why is it important? As in the beginning of the session, I told you about editing a PDF, a Word document. So if let's say you're creating a book and you make a mistake. Now in older times, there was typewriter. You could not rectify your mistakes. Now we have Word documents. You can rectify your mistakes, right? So the simple thing is CRISPR can edit genes like that. Like Word document can edit text. Gene editing can be done by CRISPR, like already mentioned. So this is how we can understand how genes work. Right? We can understand how genes work. Why? Why? Because we can turn them on and off. We can increase their expression. We can decrease their expression. All right. So this is how CRISPR is used. So scientists are using CRISPR to potentially cure genetic diseases by correcting faulty genes. Right? So example is taken, usage is taken. So gene editing, turning gene on and off. Right? Let's move ahead. How was it found? Now there was a scientist who was studying bacteria, right? Francisco Mojica in 1993. He proposed that CRISPR is something which is a genetic memory for immunity, right? But a breakthrough. Now this was just told that it happens in nature. Francisca only told that it happens in nature. But in 2012, a group of scientists, they published a paper in which they told that we can actually take this natural process and we can transform it for our own usage to do genetic editing, right? So, and in 2020, they got Nobel Prize for it. So, Jennifer Doudna and Emmanuel Carpentier, they were awarded Nobel Prize for adopting this from nature for genetic purposes, genetic editing purposes, right? So in summary, let us see in a very, let's say, informal way what these things mean so that you can remember them easily. So Cas9 are the scissors, right? PAM is the land of enemy. You identify and you destroy it. RNA is the GPS. You use it to find the enemy's land. And CRISPR is genetic information of the virus, right? Now let us move forward. How does CRISPR Cas9 gene editing works? Till now we understood the process that is going on in the nature, and briefly we have discussed here and there genes, right? And it is being adopted for gene editing. But now let us see quickly, because we have already understood the process, how it has been adopted for gene editing. So the first thing, scientists create a piece of RNA, they program it to find the gene they want to edit, right? They insert in it a guide sequence and that attaches to a specific target in the cell's DNA which they want to edit, the target DNA. Now Cas9 will follow, it will follow the RNA like the GPS, okay, like we follow Google's map, right? So it will follow the guide RNA to the location and it will cut across the DNA. At this stage, of course, since something is broken, the cell will try to repair itself, right? It will try to repair the DNA, and here scientists will do some things which we'll see. There are different processes, and that is how they can turn gene on and off and change its expression, right? So once the DNA is cut, researchers use the cell's own DNA repair mechanism to add or delete the pieces of genetic material, which I have already explained, or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. We'll see everything, right? One thing I want to mention here, CPF1. Now, like I told you, UPSC cannot ask very difficult things, but they ask related things. So Cas9 is famous. Other things are also used instead of Cas9. One of them is CPF1. CPF1. It is also important, hence I am discussing here. UPSC can simply ask, what is CPF1 related to? They can ask genetic editing, GMO, and stuff like that. Okay. So Cas9 is the enzyme that is used most often. Other enzymes are also used. For example, CPF1. CPF1 is simpler and smaller, but it cuts DNA in a different way. But that is all that we have to read about it. We are not going to see the difference between CPF1 and Cas9 because that will go very technical. And of course, it's not going to be asked. I am very sure about that because of the technical nature of the difference. So now let us see diagrammatically. Now these are very interesting. Okay. So CRISPR methods and techniques, very interesting. I'll not write anything, right? In the sense that I have not made any points because we have already explained things, but I will show you via diagram how things are done. So there is a guide RNA which we have discussed, which tells which gene we have to go to. Cas9 is the scissor, and here you can see scissors are made. Okay, scissors are made. Okay, so there is a PAM sequence. Now it is cut, right? So whenever it is cut, there is a double cut over number 1 and number 2, and there is a gap. Now this gap is going to be filled, and here scientists try to change things. Gene knockout and gene knockdown. Let us see the first two processes. Gene knockout means, before explaining, I think many of you would have seen this word used in sports, right? So knockout basically in sports, what it means, the team or the player is permanently out. Similarly, in genetic knockout or gene knockout, the gene is permanently switched off, right? We'll see how, and that is all it is doing. It will turn it off. So what happens? DNA of the cell is changed in such a way that the gene's expression, that means whatever the gene does, it stops completely. It is permanent and complete. That is why it is called a knockout. So CRISPR Cas9 is guided to the specific gene. It will make a cut in the DNA, like we have seen here. It will make a cut in the DNA, and the natural healing process of the DNA or the cell's DNA, it will start. But some issues happen, by which I mean is something is added or something is deleted while curing the gap, because of which the gene does not ever function properly, right? So for example, Lakshmikant has a chapter on parliament. It's a very huge chapter. So in that chapter, if let's say somebody comes and secretly tears off some pages, you will not be able to understand. So that book will be a waste for you, that particular book, right? So this is how a gene also becomes basically useless in expressing its capability. And the fastest method currently is CRISPR Cas9 to do this gene editing, right? So what is the usage? Drug discovery, drug screening, and disease modeling. This technique is used to understand how a gene behaves in diseases. Next thing is gene knockdown. Now knockdown means, have you ever seen a boxing match? Somebody is knocked down, right? So if they fall on the floor and get up again, that is basically a knockdown. It's not a knockout. So in knockdown, the same process is done. Scientists cut it, it repairs, but the point is it is done in such a way that it is not permanent. Okay, same thing I have written. It will survive the knockdown event. They can recover and eventually begin to express the gene as before. Okay, one tip I like to give you here at this point. See, Cas9 is not for editing. Cas9 is not for editing, but it cuts. It only cuts. But we have figured out a way, scientists of course, scientists and researchers have figured out a way to use this process by Cas9 and edit genes. Okay. So let us see what happens. So in knockout, what happens is, first it is cut, right? And the next part is the insertion or deletion happens. Now why insertion or deletion? Like I told you, when the natural healing process happens, the cells they don't repair in a perfect way, and they have something which is added or deleted, which is called indels, right? So in and del, indel. So insertion or deletion, because of which the gene will not function properly ever. That's all. Okay. Now, what is CRISPR knock-in? Now we can guess this. I think you can also guess this. Point is, knockdown is switching it off. Knock-in is inserting something in it. This is like Lakshmikant book is updated, something new is inserted, and gene will function properly. So process is, after the Cas9 cuts the DNA, scientists can introduce a new piece of DNA. Now this is the update, and technically it is called a template. In the previous process, the DNA was not given anything to cure itself. It was doing just by its own natural healing process. But in this one, in knock-in, what we do is we give it a template. Okay, you should repair the DNA like this. That's all. And then it does it perfectly. So piece of DNA that gets incorporated into genome during the repair process, right? Why? Because this process is the one which allows insertion of new DNA, new genes into the DNA, right? So you can see here, knock-in sequence, it is inserted. Let us see the bigger diagram. So here the cut is made, right? And this is the donor DNA, the whole thing actually. The whole thing is donor DNA, but this is only is taken, and with precise editing, you can have the new DNA in the older DNA, right? Next is gene manipulation. Till now we have done editing, on and off, temporary on, temporary off, permanent off, and insertion of something new. So CRISPR Cas9 can also be used apart from delete and insertion. It can be used for manipulation, which means you can, let's say there is a gene for height. You can make it in such a way that height increases, or you can make it in such a way that the gene will express itself lower and height will not be as, let's say, the parents. So gene can be manipulated in this way, right? So it is called as activation CRISPR A, CRISPR interference I. So CRISPR A increases, that means it upregulates the expression of the gene. CRISPR I can repress the expression of the gene, right? I hope this is clear. Let us compare it now with other editing tools, like why CRISPR Cas9 is particularly used so much, right? We'll see it very quickly. It is most efficient, and you can customize it very efficiently, okay, as compared to other existing genome editing tools. It is independent, it has independent cleaving ability. This means it can cut on its own. As we have seen, Cas9 is basically the scissor. So while doing gene editing, we don't need to give Cas9 anything else as a support to cut something. Cas9 itself is a scissor, right? So that is a great thing about it. Guide RNA customization. CRISPR Cas9 is, easily paired with any custom design RNA. That is also benefit. Cas9 CRISPR process can quickly follow a guide RNA, and that is also important because we can program RNA, but Cas9 should also follow that RNA properly. Availability of guide RNA sequences. Till now, we have done a lot of research and we have created a lot of guide RNAs. So CRISPR Cas9 becomes more favorable to use. Simultaneous targeting of multiple genes. I think this is self-explanatory. Multiple genes can be modified, and this is a fantastic advantage. All right. So these are the features which makes Cas9 CRISPR combination one of the best that can be used today. Right. Now the current affairs news which I had mentioned earlier, since this is a concept behind current affairs. So concept has been discussed. Now the current affairs news that made us discuss this topic with you is this. CSIR scientists, right, Indian scientists have built a breakthrough gene editor. Now what happened was, let me explain the situation. CRISPR Cas9, yes, it is being used a lot, but it has a issue which is called an off target issue. That means sometimes it goes to a different target, right? Now, this is happening a lot when we use Cas9. SP is the bacteria, Streptococcus pyogenes bacteria. Cas9 derived from this bacteria has this issue. So obviously scientists thought, let us use some other bacteria, right? Maybe it will be more efficient. So they went to another bacteria called Francisella novicida bacteria, and here it is called FN-Cas9. Now it is precise, but it also has some low efficiency. It is better than Cas9 from SP bacteria, but still has some issues. So what the scientists at CSIR did, they modified, they enhanced the FN-Cas9, right? So it is now called EN-FN-Cas9. That means enhanced version of FN-Cas9. So this is the breakthrough. Now why it is important? First of all, of course, it's an indigenous development, and this is part of the mains syllabus. Secondly, this will make these therapies cheaper, because these therapies, whatever are approved, are very costly and beyond the reach of common man. Okay, so this is the benefit for India. Now let us see the applications, and since this is a concept class, and we have limited time, rather than discussing all the applications and the theoretical ones, let me just share with you what has been done till now, which is a beautiful example of what has been done, and that will also tell you the capacity and the future of CRISPR-Cas9. So it fixed a disease in a gene in viable human embryos. So in embryos, this was changed. What happened was, there was a mutation, that means a change in a gene, right, which caused the heart muscle to thicken, and which led to sudden death in young athletes. So this was changed, right? Genome editing used to create disease resistant rice, and you can understand how important this is for country like India to ensure food security. Okay. Next, there was a lady, she was treated with genetic CRISPR-Cas9 editing, and she had sickle cell anemia. So she was the first patient with sickle cell disease and first patient with a genetic disorder to be treated with CRISPR-Cas9 gene editing, and it turned out to be very successful. She is alive right now. This was done in 2019, right? Finally, FDA, this is again in news, was in news last year. FDA, which is the drug regulator of USA, they have approved gene therapies to treat patients with sickle cell disease. One is Casgevy, other is Lyfgenia. The Casgevy one is the CRISPR-Cas9 gene therapy, right? So this was in news. UPSC can directly ask the terms and ask you what is it related to, right? So that is all for this session. If you have any doubts, please let us know in the comment section. We'll be happy to resolve it for you. All right. Thank you for your time.